In a previous video, we discussed some data from a study looking at alcohol and its effect on muscle protein synthesis, or the production of new proteins for muscle growth. The data was interesting and offered some behavior-changing insight, but we didn't discuss the mechanism by which alcohol inhibits protein synthesis. So let's discuss that now.
Learn Your Body, a science-based education.
Some of the information I'll be presenting to you now is based on the study I dissected, showing some key pieces of scientific data, and that study is linked to this content for you, along with my notes. That said, we discussed last video that alcohol can reduce protein synthesis and seems to have an effect on a variety of molecules in the muscle cell that control protein synthesis. Again, that's the process by which we produce new proteins to grow our muscles. Within the muscle cells, there are specific molecules like mTOR that control activation of protein synthesis. This is done by tagging mTOR in a particular way. This tag, called phosphorylation, either stops mTOR from working or stimulates it to work. If stimulated, mTOR will tag, that means phosphorylate, another protein that interacts with protein synthesis promoting molecules that activate protein synthesis machinery known as ribosomes. There is significant complexity here when discussing these molecules like P70S6 kinase, 4-EBP1, eukaryotic elongation factor, and, well, others. We won't discuss them in detail here, but the point is, mTOR modifies these downstream molecules by tagging them or some other alteration, and then these molecules stimulate ribosomes, the protein production machinery, to stitch together amino acids in the cells to generate proteins to make muscles grow. Now, how does alcohol affect this process? All right, bear with me. We're talking molecular biology for a few moments. If we have an mTOR molecule and we have downstream molecules and upstream molecules, meaning the downstream molecules are what mTOR controls and the upstream molecules are what control mTOR, we've discussed some of the downstream molecules generally, but we're interested in how alcohol affects the upstream molecules. So, mTOR is activated by its association to a protein called Reb. Reb uses cellular energy in the form of GTP to function because it is known as a GTP-ACE protein. So if GTP is bound to Reb and Reb is bound to mTOR, mTOR will downstream signal to protein synthesis. However, there is a molecule that controls Reb, and that molecule is called TSC. So if TSC is active, it will bind Reb and, for a lack of better words, transform the GTP to a GDP, a different molecule. In doing so, Reb is no longer active, so it no longer activates mTOR, and protein synthesis reduces. Great. TSC controls Reb, thereby controlling mTOR. Now, allow me to introduce one more molecule, Red. Alcohol seems to increase the amount of red production by the cell, and red stimulates TSC. Clearly, this then means more TSC activation by red leads to less Reb function on mTOR and less downstream protein synthesis drive by mTOR. It seems that alcohol may stress the cells in such a way that they produce too many misfolded proteins, or junk proteins, creating cell stress that needs to be fixed. So the cell responds by increasing the production of red, specifically, which then reduces mTOR activity, reducing the protein production to reduce the production of more misfolded proteins, until the cell can clear them out via cellular degradation machinery that we won't go into. That all said, this is a single mechanism of likely several that mediate this whole process. Red is but one protein, the molecule that is postulated to play a role, but others like Raptor, AMPK, and others likely all play a role in this as well. So don't be deceived into thinking this one pathway I've outlined here is the end-all be-all. Yet, I hope this provided you with some insight on how alcohol leads to a reduction in muscle protein synthesis. And with that, I hope to have the pleasure of speaking with you in the next one. Cheers.